Hello everyone, we are solving question number 39 of GATE 2016. It says, which one of the following statement regarding G protein is incorrect? First, GDP is bound to G protein in resting stage. Second, GTP bound alpha subunit cannot reassemble with beta gamma dimer. Third, all G protein are trimeric. Fourth, activation of G protein may result in activation or inhibition of the target enzyme. So to solve this question, let's understand how G protein works. So, uh, so the first step in G protein signaling is that GPCRs undergo conformation changes upon ligand binding. So once the ligand bind to the GPCR, it activates the pathway. So once the ligand bound, so if you could see here, GDP is bound to G protein in resting stage. So the first statement is correct. Once the ligand bound, G alpha subunit exchanges GDP to GTP. So there is an exchange of GDP to GTP. And once this exchange happened, now GTP bound alpha subunit cannot reassemble. So the gamma and beta dimer is removed. And if you could see here, this beta dimer is removed. Now it cannot reassemble with beta gamma dimer once it is bound to GTP. It will always reassemble with them if this GTP is converted back to GDP. So second statement is also correct. Then they are saying uh, so once there is a GTP, so step 3 is activate adenyl cyclase. So adenyl cyclase is activated that convert ATP to CMP. So ATP to CMP, CMP is a secondary messenger. It further activates, elevate CAMP level, then activate protein kinase A. PK is inactive. It is active once CAMP levels are elevated. And once the PK is active, PKA, that is protein kinase A, it phosphorylates several enzyme and transcription factor downstream. CAMP response element protein, so for example, it binds to PK binds activate the CREB transcription factor. So CREB proteins are activated, causing gene expression that mediate cell growth and differentiation. So this is how GPCR, or you can say G protein work. So coming back to the question, this third statement says that all the G protein are trimeric. We cannot confirm that by seeing this. So it might be wrong, but let's see the D option that says the activation of G protein may result in activation or inhibition of target enzyme. So yes, once the G protein is activated, CMP levels are elevated and there is an activation of PKA enzyme. So PK phosphate activation of protein kinase A. So enzymes are activated as well as inhibited. So D is also true. But let's come to the C, which says that all the G proteins are trimeric. No, RAS, it is also a G protein, but it is monomeric. So RAS protein is a G protein and it is in monomeric stage. So option C is wrong, and we have asked to select incorrect statement. So answer here going to be C, that all the G protein are trimeric. So here I have given you a list of G protein and their secondary messenger pathways. So if you see, the first one is GS, that has alpha as a action subunit, and its function is to activate adenyl cyclase and activate calcium channel. Then we have GOS, that is uh, for the olfactory G proteins. Here also you have alpha subunit as an activation subunit, and it activates adenyl cyclase in olfactory sensory neurons. So G-olf is related to olfactory sensory neuron. Then we have GI. GI, here also we have alpha subunit that take the action and it inhibits adenyl cyclase. So in case of GI, alpha subunit inhibit adenyl cyclase, but its Beta gamma subunit activate calcium channel. So in case of GI, alpha inhibit an adenyl cyclase, but beta gamma activate calcium channels. And coming to G naught, that is here beta gamma subunit activates calcium channels, inactive potassium channels, inactivate calcium channels, and alpha and beta gamma subunit together activate phospholipase C beta. So for G naught, these subunit acts like this. Then we have GT, that is for transducin. It also have alpha as an action mediated subunit, and it activates cyclic GMP phosphodiesterase in vertebrate rod photoreceptor. So this happened during transducin. Then we have third family, that is GQ. It its action is also mediated by alpha subunit, and it activates phospholipase C beta. So these are the function of different type of G protein and their secondary messenger pathways.